Networking in Coimbatore. I also welcome all the faculty members and students who have joined the session. Now, I request Rutuja Shinde ma'am to introduce the speaker. Over to you, ma'am.
Thank you, Shraddha. Am I audible?
Yes, ma'am.
Yes, ma'am.
Yes, yes.
Good morning to all. I, Rutuja Shinde, Assistant Professor of
Hello, ma'am.
Sihagar College of Pharmacy, I welcome you all for today's session. It gives me an immense pleasure to introduce today's guest speaker, Dr. Harishchandar Anandaram, who completed his PhD in bioengineering and MTech and BTech in bioinformatics from the Satyabhama Institute of Science and Technology, Chennai in 2020. His bachelor's and master's thesis, he worked on analyzing resistance in HIV protease inhibitors based on molecular mechanics and machine learning studies, a collaborative project with IIT Madras. In his PhD thesis, he worked on pharmacogenomics and mRNA regulated networks in psoriasis, a collaborative project with Georgetown University, USA. His thesis illustrated a multidisciplinary approach by combining computational biophysics and molecular biology machine learning. While doing a PhD, he had the opportunity to collaborate with international researchers and have publications in reputed international journals in bioinformatics and system biology. He has received the prestigious Young Scientist Award from the Melinda Gates Foundation for his research abstract on the implications of mRNA dynamics in infectious diseases. To date, he has reviewed more than 200 manuscripts in system biology. He is currently working on predicting novel lead molecules and biomarkers using computational techniques to target inflammatory pathways associated with infectious and autoimmune disorders. With this brief introduction, I would like to welcome Sir and start with the session. Please, sir.
Well, thanks for the brief introduction. Well, I hope my voice is audible and screen is visible. And today's topic, pharmacogenomics and microRNA regulated networks in psoriasis. Just to give a brief intro, psoriasis is an autoimmune disorder, and both pharmacogenomics, that is why some drugs are working nicely for one person and not for the other person. That's based on the SNP markers, that's a single nucleotide polymorphism. Pharmacogenomics is triggered. And then with respect to the small RNAs, microRNA served as a very good biomarker for cancer. And in autoimmune also, there are a lot of good signatures. And even some of the microRNAs predicted from our side is also in the phase 2 and 3 clinical trials. And based on these leads, my thesis was guided by Dr. Daniel from Satyabhama University and the examiners, Dr. Jayakantan, Professor and Head in Bioinformatics Department at Satyabhama University, and Dr. Karthik Ram, my Indian examiner. And why I'm taking psoriasis? Because worldwide, if you consider, psoriasis is twice more common in males than in females. And then till now, there is no proper reliable information. They are saying some alternative pathways to cause psoriasis, but this is the exact reason till now people don't know. That is the main problem with the autoimmune disorders. The exact mechanism and the reason is not yet completely known. Only the alternative pathways are known. So how we are going to go a little deeper based on the alternative pathways and the related informations is a part of a study. And then in India, you can see from 0.44 to 2.8%, the rate is increased in from various hospitals. And the epidemiology journal also says the same fact. And why the complexity occurs in most of the autoimmune disorders like psoriasis is because you have both genetic and environmental factors working dynamically together in case of causing autoimmune disorders. And then the inflammatory pathway triggering is also not based on the single receptors, it's based on the multiple receptors. And what were all the key aids when the studies were enhanced? When you look a little deep into it, in case of the development from the pre-genomic era to the post-genomic era, the level of information is very high. In pre-genomic era, we had only DNA blotting or FLP maps, some basic linkage to human genome. But in case of the post-genomic era, you have the haplotype mapping and the genome-wide association, so that the multiple locus were identified.
Sir,
And
sorry
to consider
to
the
disturb.
recent developments, yeah, yes, please.
Can you just put the slides on the presentation mode?
Okay, fine. I'll do that.
Yes,
I
yes,
hope
sir.
now it's
Thank
clear.
you. Yeah. Thank you.
Okay. So, in the post-genomic era, you had a lot of SNPs being discovered due to the HapMap and GWAS projects. And those SNPs gave us a lot of information related to fine-tuning studies. So if you consider the genome-wide association studies, which was published for 221 tribes, a lot of additional locus information we got. Initially, people were working only on the HLA markers. Now we have got more than 100 markers to work. You can see it is not only done for psoriasis, for various studies. So with respect to psoriasis, we got basic information, and from the basic information, the additional information in our research were also triggered and executed. So in the abstractors, we need to identify a computational approach. But when I was starting my PhD, there were only one or two approaches. We have given 4 alternative approaches to predict the markers. And we have found one particular mRNA marker, which is highly compatible and can be given to the wet lab people for further experiments based on the binding compatibility and the level of totipotency. And objectives, we were not followed a single approach. It's a multiple approach like top-down, bottom-up, direct and indirect. In top-down, we took data from PubMed, OMIM, and DisGenet. In bottom-up, it's the RNA 6 studies which we have performed for the control and treated sample. And then the pharmacogenomic based linkage pattern analysis. And finally, the transfact, that is the translation factor associated matrix were created. And then the network biology based concepts were executed to predict the marker. So literatures, when I was starting my PhD, there were very few literatures on biomarkers. Now you have a lot of literature. A daily basis, lot of works are being done on biomarkers today. And Liu was the person who gave the first transcriptomic file, and from that we extended a part of the work. And you can see when it comes to the approaches, only in the network analysis part and the RNA interaction part, there are some minor variations. Rest of the part is the same. And the data we have used is only slight different with respect to the approach. And you can see, though there are a lot of compatibilities for microRNA, there is no proper binding. You can see in databases, some microRNAs are related based on the sequence alone, but they don't have the structural folding and other regulatory aspects and the binding free energy to go to the further level. So those screening was done. And then it's not only the microRNA and the transcription factors present in the simulation which we work. We have also cross-checked with the experimental data of the transcriptomic profile in the human genome map also. From that also we have found the relations, and then we got the relative information. So the regulatory networks were modeled, that is the gene mRNA, the transcription factor based network. And then here also you can see very few microRNA show some proper binding. And when you consider the discussions, like most of the results we have reported were also reported by the other studies. So those markers were taken out because it's already reported. Some additional markers which we have reported, which has not been reported, were being studied further. You can see STAT2 and all already reported, and this particular mRNA which we have worked has not been reported. And in the initial assays also they got good screening, and now it's in the phase 2 and phase 3 classes of psoriasis. But in case of cancer, it's already being worked. And you can see the implication of the regulatory network from the top-down and bottom-up approach. The ACTL8 marker is yet to be explored in psoriasis, and now people are exploring in China and Japan related countries for further works. And you can see it's not only the regulatory network we are considering on the protein-protein network also, and how the upregulation and downregulation triggers the associated process. And from then we got the gene ontology based factors. And you can see, though we have a lot of pathways, the cytokine chemokine receptor interaction which shows the higher p-value, and that is taken as a standard. And then the mathematical modeling was executed. So we have formed a first order differential equation, because in the initial rate, the synthesis is related to the regulation. Based on that, we formulated the equation and then modeled the network and got some unique hits. WIF1 were to be explored in psoriasis, which we have reported first. And then the related networks of STAT3 were also executed. And you can see only the one which had proper binding and scoring, which can form the canonical triplex and the duplex, were taken. And finally, they were cross-checked with the human genome browser and the transcription factor-based interactome studies. And then you can see the various alternative pathways which can be used in modeling psoriasis, and why the experiment fails in initially, how the alternative pathway can enhance to get a good experimental results were also triggered. And you can see there are some missing connections, and how to reconstruct the pathway was also being analyzed together. And it's not only the basic expression, we have worked on even on the co-expression. Sometimes even the co-expression, though it's not initially expressed in the initial stage, some other factor binding may lead to the expression. Those kind of analysis were also done. And then we reconstructed the model based on the co-expression also. And as you can see, there were very few alleles which is based on the SNP, which shows some proper interaction for the markers. So we have taken those promoter and the transcription factor binding site and associated with the genes already in the pathogenesis and the genes which we have worked on, and finally got the comorbidities associated with the disease and the relative network also. Though comorbidities don't give the complete picture, it gives you an initial picture so that in future the triggering might happen. That also we have modeled. It's not we have modeled a single system, multiple systems with a proper reconstruction. And then the genes which are co-expressed in psoriasis and rheumatoid arthritis were also screened, and the ones which are unique to psoriasis were taken from arthritis, because a lot of similarities will come, and that should not end in false positives. So the cellular communication based model subset also was taken. And then the genetic variations involved in psoriasis and cell-to-cell communication were also analyzed. And finally, we got a simple transcription based network for the MTHFR gene in the pharmacogenomic approach. And the activation of MTHFR is by AHR. It's a unique finding which we got it first. And then the same regulatory network, how it is modified with respect to additional results were taken. And then the hubs were also calculated. That is the genes which are related to the regulatory. And then if you consider the implication, very few form the canonical triplex from the duplex. And one more interesting is that HTL1B infection also showed a very good relation next to the cytokine chemokine receptor interactions from the pathway. And then the modeling made a little easy. Epidermal hyperplasia is the major condition which is happening in most of the psoriasis. So the TNF alpha related binding chemokine released system was taken initial, and then our reconstructed pathways were also used in the modeling. You can see the type of modeling which we have performed from the initial to final concentration, how the influx and the related metabolic flux varies in the removal as well as the association. And then we used MATLAB for modeling the interleukin 23 mediated pathway by using the ANFAS method. While doing the combinatorial analysis, we have got 71 genes as unique from the total number, and we have reconstructed the regulatory network. And we have also analyzed the subnetwork based on the final regulation to get some initial conditions for the inflammation-mediated autoimmunity. And then we have also used the regulatory pathway analysis to identify the additional factors which trigger psoriasis. And MAP kinase and related pathways gave us good results. So we have got a lot of data. And finally, we have done a cascade analysis also. Because we have multiple cascades. From these multiple cascades, how the disease association occurs. Because it's not multiple cascades, multiple times you end up in a disease, very few times you end up. So from the multiple cascade to the initial cascade, we got the disease-related association, and then the matrix associated for the disease. So the one which are having more disease-related associations were taken, and then the final pathway reconstructed network was formed. And we have identified the hubs within the final reconstructed network for the unique mRNA of our study. And then we reconstructed the final network. And then we have also looked for the biological process, that's the signaling pathways and the initial basic pathway. And we have tabulated the results. So to summary, the study has given us only a basic understanding to deciphering psoriasis. Furthermore, putative targets are required for the further explanation. But the pathogenesis is explained a little more in our study compared to the initial studies. So a lot of biological experiments are still required to validate our study. The scope is we need to work on the simulation of regulatory networks from mathematical model to identify the significance. Already a lot of works are going on all over the world to identify a biomarker related to it. And this was the recent publication. Initially, we got it from an Elsevier journal. And these are the related journal publications. And these are the conference national and international level. And these are some of the key findings from various journals we have reported in 2018-14. And these are the MIRMAP results, that is the binding site association and number of binding sites associated with the RNAs, and all those models were analyzed. And then the queries from foreign examiner, like this looks like a computational study, will it help? Because when we were starting the study, there were no proper pipeline, one or two are there, but we have developed this pipeline for regulatory analysis, and already it is cited by many papers, and a lot of works are being going on, and the sample size is less. And how do you going to extend means we'll need to do it for 10 samples, because what we have done it for only for 3. And when with respect to the biomarker, how it will be means it's a therapeutic marker associated with C67T polymorphism. It's a finding which is reported in 2016 because we reported in 2014. After that, some relation came in another journal. So that, and then the mTOR signaling in phosphoinositol kinase 3 and the AKT pathway is also related to the signaling. So whether the model can be directly applied to the other diseases, yes, it can be applied because all factors were included. And what's the purpose of differential equation in the sense? Then only the transcription factor based repression in gene regulation will be analyzed. And in case of the IL-23 induced hyperplasia, the psoriasis activation through the cytokine chemokine receptors, that is the CCR5 receptor, can be explained by the NF-kappa B. And with respect to degree network parameters, though we have a lot of parameters, degree showed a lot of promising in case of the hub analysis. And with respect to the extension to other diseases, yes, it can be extended, and this can become a pipeline for mRNA-based gene regulation. And the most promising target, as we have reported, MR125A3P, a therapeutic marker. It was explored in cancer initially, and now people are trying to explore in psoriasis also. And once the final phase 3 is completed, it has got a good potential in the market also. And it has got some factors in basic assays to understand the pathogenesis of psoriasis. And the forum is open for discussion. I thank the people for giving me an opportunity to present the work. Okay, I'm done with my presentation, and queries will be entertained. Thank you.
Thank you, sir. If anyone has any question, please feel free to ask.
Thank you, sir. Thank you for the presentation. I have got one question.
Please, sir.
So we have got a couple of groups at our college as well who are working on cirrhosis. One of the thing while doing my postdoc, I came across with one of the treatment called PUA treatment, which is useful in cirrhosis.
Yeah,
So my
some
question
people.
is,
Yeah, please.
So my question is that, yes, some people are going to have difficulty when they use this PUA treatment because it is a combination where the patient is exposed to UVA and there is a chances of leading towards cancer. So basically we were working on that. But then if there is a treatment already available, what means how can we correlated that with this work? So that's the question from my side.
Yeah, yeah. Thanks for the good question, sir. Because as far as I know, it's only the methotrexate-based ointment that is showing some good results from the patients, and other sheet treatments are showing some relatively positive results, but not like methotrexate. And even people try to use the analogs of vitamin D. That also is not working completely, and some vitamin D related supplements are working. So we need to have a perfect biomarker, actually, a therapeutic biomarker to address the issue. Then only it can be completely fulfilled. Some groups are targeting some peptide biomarkers. I hope I've answered your question.
Yeah, thanks. I mean, in my group, we actually just starting recently. So as I said that our main aim is to target we are actually targeting cancer, and we're looking at the DNA damage and DNA repair. So as I said, I came across with the SPOA treatment, which is a combination treatment where the patient is given a drug called Sorelin 2 hours before the treatment with UV rays. And what is happening if the person is having a white skin, there is a chances that the person might lead towards skin cancer. So we are actually one of my students, she is working on this now, where she will be exposing the animals, and we are going to look at this. So maybe I think we can actually possibly have a collaboration and can work with this.
Sure, sir. I'm happy to
That's
collaborate.
that's the one of the thing. And second thing is we also got a, collaboration with a company named Leo Pharma, which is
Okay,
based
good.
in Europe, and they do work on this type of product, mostly about the skin product, dermatologic product. And we are part of the open innovation platform of that company. So under that, what we normally are doing that we are sending our formulations or extract to that company, and they are testing in that pipeline. So last time when I actually sent them the sample, they tested on the cirrhosis. So maybe that's something can be helpful for you as well if you're planning or if you're
Sure.
just planning towards. Yeah.
Yeah, sure, sir. It's more helpful for me. Thanks a lot for the information. Happy to
Yeah, and
collaborate.
like if you're
Thanks a lot.
just if you're just planning for molecules, we have got department where we can work on the formulations, and then we
Yeah,
can
synthesis part
go
and all.
through that. Yeah, synthesis and also formulation part of the molecules which we maybe identified from the natural sources. So
Okay,
that's something we can
thanks.
do.
You
Yeah,
work on
any
peptides also,
question?
sir?
No, we actually work on small molecules.
Okay, sir.
No, we're not working on peptides. But maybe
Okay,
from
sir.
our college, if somebody is working, then definitely I will let you know.
Sure, sir. Small molecules also we are working, and
Mhm.
simultaneously we are working on peptides also.
Okay.
One of my student is working on peptides. They started a company recently in
Mhm.
collaboration with Europe for peptides.
Okay, that's good.
Thanks.
Any questions from the students, faculty? Okay, Shraddha, we can move to next part.
Okay. Now I request Abhishek More to propose the vote of thanks.
Good afternoon to all. I feel honored to propose vote of thanks on the behalf of Siagat Technical Education Society, Pune, for this session. My heart fills with lot of gratitude and respect for our guest speaker, Dr. Harishchandra Anandram, for not only sparing their invaluable time for us to gracing this session, but also enlightening us with their commendable talks on pharmacogenomics and mRNA regulated networks in psoriasis. Thank you so much for your guidance. We are thankful to our founder president, Professor M.N. Navle sir, founder secretary, Dr. Sunanda Navle ma'am, vice president admin, Mrs. Rachana Navle Astekar ma'am, vice president HR, Mr. Rohit Navle sir, for their motivation. I am running short of words to express my humble thanks to our president, Dr. Rajesh Patil sir, for arranging, planning, and making this meeting successful. Your support and motivation lead us on the path of success. I would like to thank Dr. Praveen Bade sir and our teachers for their active participation in making this meeting successful. Once again, thank to one and all who contributed in this meeting. Before finishing this session, everyone please turn on the cameras for a group photo. Thank you. Please everyone turn on their cameras for a group photo.
Thank you.